Guys, Anna here, and this is a part 2 video in where we're going to be discussing the sliding filament theory of muscles. So please grab a piece of paper so you can make the notes with me alongside. The only thing I ask in return is please subscribe to my channel and like this video, and then let's get into it. So I think it's quite important that you watch a part 1 video about the muscle sarcomeres and the different structures as a summary and a preview of this video. If you watched it already, then we can just jump straight ahead and start talking of the structure of actin and myosin. So let's have an overview first. As a reminder, skeletal muscles are made out of muscle fibers, and muscle fibers, each of them is a singular cell. And within that cell, there are going to be many structures, again, which you can watch in a previous video, but they will be also consistent of myofibrils. And myofibrils consists of two filaments called actin and myosin. And at the moment, I'm drawing the myosin filament, which has globular heads as well, which will become very important in this video. And also myosin is referred to as a thick filament. The second filament that is part of the myofibril is called actin, and actin is another biological polymer, and it's referred to as a thin filament. And basically what I've drawn here is a quick preview of the sarcomere, but a more detailed structure, again, please watch my previous video. And what happens during the sliding filament theory, as I've showed with arrows, is they're going to be going towards it. So it's going to be shortening during the muscle contraction. So to understand this actin and myosin cycle, we need to look into more detail for the structure of myosin and actin. So if we draw actin first, which is a biological polymer, and it consists of two helices. And I think the best way is to actually have a little zoom in box and see what goes on in more detail. So if you look at other different pictures provided online, you will actually see that actin they draw them as like almost beads on the necklace, and each bead represents a monomer that has a myosin binding site, which I'm going to label here in purple color. These myosin binding site are covered by a different structure, a different molecule called tropomyosin. And it's usually represented like as a line like this, but obviously it's going to follow the structure of the actin. And furthermore, this tropomyosin is bound by troponin, another molecule here, which almost acts like a stamp. So it ensures that tropomyosin does not dissociate from the actin myosin binding site. Okay, so, and then just as a reminder, if we draw the myosin here, the thick filament, and the globular heads as well, myosin globular heads, so they will eventually be complementary to the myosin binding sites, but currently they cannot bind to those sites because they're covered by tropomyosin and troponin. So the first thing to understand is how actually these sites become available. Brings us to the next subheading, the actin and myosin cycle. And just as a reminder, what I'm going to do is I'm going to copy over this diagram and bring it in the corner. So as we write notes, you guys can actually have a look at this in a more detail because it will get quite complicated. So there are a lot of notes involved for this topic, but again, this is like a model answer that you can just learn for the exams. So let's start by saying that action potential arrives at the neuromuscular junction. So via the T tubules, again, remind the T tubules that the extensions of the sarcolemma, so the action potential will travel down those, and then it will reach the sarcoplasmic reticulum. And sarcoplasmic reticulum, it stores calcium. So as the action potential arrives, those calcium ions will be actually released from the sarcoplasmic reticulum. Interestingly, these calcium ions, they will actually then go and bind that protein troponin, which will cause the conformational change of troponin. As you can imagine, calcium ions, they're positively charged, so it will have effect on the protein. And what happens, it basically then, that conformational change will also result in the conformational change of tropomyosin, and therefore tropomyosin is going to be able to move away and actually make myosin binding sites on the actin filament exposed and ready to bind to the myosin globular heads. So they become available for that cycle to start, so between the actin and myosin. As this is quite a complicated topic, it's really important that you guys draw doodles alongside and diagrams to help you understand this topic. So again, you can just draw a couple of actin monomers near the myosin filament, and you can just move the tropomyosin away and actually show that those myosin binding sites, they're actually now available, and eventually the myosin globular head will bind to those sites. Okay, so let's break it down and say that this is like the first part of the actin-myosin cycle. And now let's kind of see what actually goes on between the cycle of the actin and myosin. Just as a reminder again, if we draw the actin with the myosin binding sites like this, and we're going to draw the myosin filament now actually binding to those sites available. So globular heads of the myosin molecule filaments bind to the actin myosin binding sites, forming the cross bridge between the actin and the myosin filaments. This formation of cross bridge causes the myosin head to bend, releasing ADP and inorganic phosphate, which results in the pulling of the actin filaments towards the center of the sarcomere and thus shortening it. And this is important because this is what indicates that the muscle is actually undergoing the contraction. So the key thing to note here, so this is going to get quite complicated very quickly, as you can see, but it's important to basically associate different states of either ATP or hydrolyzed ATP with what is going on here. And I think this is actually the best way to memorize it this way and understand it. Okay, so what then happens next is that the ATP binds to the myosin head, causing a conformational change, which basically results that myosin head to be released from the actin and actually not be bound to it any longer. Furthermore, then the enzyme ATP hydrolase comes in and hydrolyzes this ATP into the ADP and inorganic phosphate, which then causes myosin head to move to its original position. And basically, this is how the cycle is actually then going to repeat itself. What then happens is basically, so it's unbound, and then the myosin head, it then goes and binds to the new binding sites on actin filaments, and which basically will be closer to the Z line of the sarcomere. If this is repeated several times, then the sarcomere is going to get shorter and shorter, and then this will basically result in the contraction of muscles. Anyhow, guys, I hope you enjoyed this video. It is a hard topic, but I've provided you with this model answer that you can then use in exams. Please like the video, and please do let me know what you would like to see in the next topic, especially now during the exam season. I'd be happy to make more videos. So good luck in your exams, and I will see you in my next video. Bye bye.